Hello and welcome back to Communicable, the podcast brought to you by CMI Communications, ESCMID's open access journal covering infectious diseases and clinical microbiology. My name is Erin McCreary, and I'm the Senior Director of Infectious Diseases Strategy at the University of Pittsburgh Medical Center in Pittsburgh, Pennsylvania, U.S. I am joined today by 4 other fantastic CMI Communications editors to discuss the clinical trials presented at ESCMID Global 2026. This is part 1 of a 2-part series because there was so much fantastic content. So with no further ado, let me introduce you to the wonderful editor team here. So first we have Josh Davis. You know him, you love him. He's an infectious diseases physician and the head of the Infection Research Program and the Huttner Medical Research Institute at the University of Newcastle in Australia. He's also a clinical trialist. Josh, welcome to Communicable.
Thank you, Erin. Great to be here, and I'm very excited to be talking about late breaker clinical trials, one of my favorite things. And I'm now going to introduce Marc Bonten. Marc is a professor of molecular epidemiology at the University Medical Center Utrecht. Did I say that properly, Marc?
Very
Utrecht.
good.
Where he is currently Vice Dean of Education. Marc's also a clinical trialist and a fellow editor on CMI Communications. Welcome, Marc.
Yes, thank you, and hi all. And I'm very, very pleased to be here again. I think it's the 3rd year in a row that we're doing this session on the trials at ESCMID. I am especially pleased to introduce Anne-Grete Märtson, who is our newest CMI Comms editor, and she's making her Communicable debut today. Anne-Grete is an assistant professor at LECDR. No idea what it means, what it stands for, but it is at the Leiden University, where she's leading the antiviral pharmacology research group. She's also a clinical pharmacologist. Anne-Grete, welcome.
Thank you, Marc, for the nice welcome. I'm very happy to be here. I'm happy to introduce Angela Huttner, who is an infectious disease physician in Geneva, Switzerland, and of course our editor-in-chief at CMI Communications.
Thank you so much, Anne, for the lovely introduction. I'm so excited to be here as well. I'll dive right in because we have so much to cover. We will start with our usual get to know you question. So for this week, if you were organizing ESCMID 2030, what city would you organize it in and why? Marc, you go first.
Yes, I've been thinking about this for a whole week now, and I made actually criteria. So it should, for me, be easily accessible by train with not too high risk for delays. There should be a reasonable chance of good weather. There should be nice and affordable restaurants. And there should be something to spend a day outside of the conference. So that brings me to either Paris or Amsterdam. Pick your choice.
Ooh, nice. But I mean, I hate to say this, but it's like often in Amsterdam, Marc.
It's
you
April.
Guys always get it.
Yeah, well,
Because they have tulips. It's wonderful.
yes, it's... If
The
you fancy
Amsterdam
tulips,
accent-
that's never thought of the tulips for that reason.
Are you
but
kidding? That was the first thing I did when it was in Amsterdam. I went up to the tulips. It was amazing.
People
No,
go for
I fully
flowers
agree.
and
Like Amsterdam is practical and easy to reach. Very practical. I think those are very practical
there's
answers.
other vegetation in Amsterdam that's attractive to people. No.
This is a PG podcast. I
Okay,
know.
sorry. Okay, we're cutting that out. All right.
No, we're not.
Oh, we're not. Okay. Next up is Josh. What's your city?
So I guess there's limitations now that ESCMID has become so gigantic. There were like 18,000 delegates this time. There's only a limited number of cities that can actually have the infrastructure to do it. So I don't know what cities can and can't, but I was going to say Paris also because I think Paris for me is a really good overlap between it's a big enough city to do it and it has the infrastructure, but it's also, a beautiful place to visit and it's romantic and there's things for tourists to do, and I'm a Francophile, so I would say Paris.
Okay, so you guys are very, very realistic people, but the entire premise of this question is totally counterfactual. So I did not limit myself to any realistic constraints. What I would do would be to have, and this is a total bias, obviously, I would have ESCMID 2030 in Italy, in Trieste, which is the city on the northeastern border of Italy. And you guys have probably heard me. I've talked about Trieste already once, at least on this podcast. So it's not realistic because I don't believe there is a convention center big enough to hold so many delegates. But Trieste is an amazing city. It's not hard to reach. The airport from Venice is not far, and it's on the sea, and it has good cuisine, and it has beautiful places to go, and it's an underrated city, which I'm very biased about because it is the city where my mother originally came from. So Trieste, Italy for the next ESCMID Global.
Can I change my answer? If we're being impractical,
yeah, we are.
I would have it in Sydney because I'm gunning for Australia to be considered part of Europe, right?
Like Canada.
We're getting more and more involved in ESCMID. We love Europe. We love ESCMID. So I want you all guys to come to Australia. Sydney's an awesome city and we can hold big conferences.
Love it.
Sydney makes a lot of sense from a logistics standpoint too. Okay, so I wrote this question, and then as you were asking it, Angela, I was like, did I think of an answer? But I did. So along the somewhat dream, because I don't think structurally it could hold it, although I do think they have a 15,000 person conference center, so just shy of what's needed for ESCMID Global. But I would do it in Zagreb. I adore Croatia. I would actually do it in Split or a coastal city like Hvar. Like let's just rent the whole island. Let's just actually don't even know if there's infrastructure on Hvar for 19,000 people. But I mean, that
again,
would be the
we're dreaming here.
Oh my gosh, to be on the sea. Oh my gosh. And in the olive oil vineyards and wineries. I mean, what a dream. But practically, I don't know. The capital might play and then it's not too far. And then if you want a vacation on the front end or the back end. So that's what I would do.
Nice. Yeah. Definitely off the beaten track. I love it. Yeah, Zagreb is a beautiful city. I don't know Split. I've seen pictures. It's amazing. You can always dream. Yeah, exactly.
And
last but not least.
Yeah, so I'm totally unoriginal, but I also thought of Paris. It fits with Marc's criteria, I must admit. But I actually thought Paris because 2020 was cancelled and it's so sad. I feel like it was sad for them. They should have another chance.
Sad for us. I still feel
yeah, but also them. I guess that they spent a lot of time organizing and everything. It was cancelled quite last minute in the end, so...
It's funny you say that because every time I think of Paris, I think of how in March of 2020, we all were like, oh, this will only last for 2 weeks. And I kept all my bookings for ESCMID Global. I was like, it'll be fine. Everyone's telling us this will be 2 weeks of lockdown and then it'll be fine to travel. And oh my gosh, how little we knew back then. We did end up canceling that trip, which was sad. So sounds like ESCMID Global organizers, you have a resounding vote for Paris. So. Okay, let's get started with the trial. So a lot of us at CMI Comms doing this podcast are trialists, are clinician researchers ourselves, are practicing in labs or in clinical care still. And so we inherently are somewhat involved in a lot of these trials, whether we intend to be or not. And so as we go through these episodes, we will, if we comment on the data for the trial, whether we're the one presenting the trial or have commentary, we will verbally state if we have any conflicts of interest. Otherwise, all of our conflicts of interest will be listed in the show notes, or if we have a genuine conflict, we simply will not comment on that trial. In this first episode today, we are each going to review one trial, and then for part 2, we'll each share one more. So listeners, you're going to get a review of a total of 10 trials, which we are very excited about. And Marc, my friend, why don't you start us off with the first trial?
Yes, let's do that fast. So I will start discussing the fast antimicrobial susceptibility testing for gram-negative bacteremia, RCT, which was presented by Ritu Banerjee from Vanderbilt University in Nashville, and which actually was in a session organized by JAMA. So JAMA organized a session on clinical trials presented by people from the United States in Europe, and they were publishing at the same time as the presentation was each of the studies in their journal on their website. So that was really an event. Now, the FAST trial. Little background on how we practice. We have a clinical suspicion of infection, we take a blood culture and usually start antibiotics, and then we wait for the results of the blood culture. If the bottle turns in positive, we make a gram stain, and then we have some guidance on how to treat the patient optimally, and then we can start by adjusting treatment, de-escalation, escalation, waiting for the susceptibility testing results, which can take more hours. Now, the hypothesis here was that if you reduce that time for waiting for susceptibility results, that will improve patient outcome. So the intervention here was that there would be direct antibiotic susceptibility testing and identification with a new tool, the VITEC Reveal, and that would be compared to the standard of care, which means subculturing the blood culture content, waiting for the bacteria to grow, and then do the susceptibility testing. Now, the potential gains are obvious. You have faster information to give the appropriate antibiotics, which could improve patient outcome, and you could have information to faster de-escalate antibiotic pressure and thereby, let's say, reduce the selection pressure for antibiotic resistance. The primary outcome of the trial was the desirability of outcome ranking DOR at day 30 after the gram stain. And the DOR is a composite endpoint with outcome parameters. I think many of you are familiar with that. And in addition to that, there were many secondary outcomes. The study was done in 7 sites in Greece, India, Israel, and Spain. And very interestingly, the study could be done as in 6 of these 7 sites, there was a waiver for informed consent. So the randomization could be done in the lab, and the physician and patients were just not informed about that they were part of a trial. I've tried to do this trial in the Netherlands, but it would not be possible without an informed consent. That's why we could never do this trial, and that's why I'm so happy that now we have the trial. In total, they randomized 899 persons, 450 to the intervention, FAST, and 449 to standard care. Now, this was a typical hospital-based population with a median age of 73 years. Almost 50% of the patients came from Greece, 35% from Israel, and just over 50% of them had hospital-acquired bacteremia, mostly coming from the urinary tract. And very importantly, this was a population in a high prevalence AMR setting. 37% of the isolates were resistant to cephalosporins, and 19% were also resistant for carbapenem antibiotics. So this is a above average, certainly for Europe, resistant setting, antibiotic resistant setting. Now, before anything happens because of the intervention, patients will be treated with antibiotics, and the initial antibiotic treatment was considered effective in about 62% of all patients, equally in both arms. Now, what did the intervention achieve? Actually, there was a higher proportion of patients on what they considered effective antibiotic therapy at 24 hours. It was 84% in the intervention group versus 75%. And that's for the total population. Now, for the cephalosporin-resistant subpopulation, the difference was larger, 82% versus 63%. And for the carbapenem-resistant subpopulation, the difference was even larger. It was 72% versus 47%. But that was at 24 hours. And indeed, there were more stewardship recommendations made within the first 3 days, and more recommendations were accepted within these 2 days. So there was definitely an effect of the intervention. However, that effect was already equaled out at day 3, because at day 3, the proportions of patients on effective therapy were equal in both groups, and also for the subpopulation. So the window of benefit of this intervention lies between 12 and 48 hours after the gram stain. And that window of benefit is nicely illustrated in the survival curves up to effective therapy. There the lines are initially similar, and then they create some space, and then they come together, and then they create this window of benefit, which is larger if resistance increases. Now the question is, what does that window of benefit create for the patient? Well, and that's, in this trial, actually very disappointing for the patients, because the DOR probability was 48.8%. And actually 50% means that there is no difference in any, let's say, in the composite of the endpoints for patients, 48.8% going from 45.3 to 52.4 actually also means the same, that there is no real outcome difference for patients. And that's the primary outcome of the study. However, there were also 27 secondary outcomes. And among these 27 secondary outcomes, there was one in which there was a clinically significant difference that benefits the intervention. And that was the proportion of patients that were still hospitalized at day 30 in subpopulation of carbapenem-resistant infections. So in 1 out of 27, that was the positive signal. Now, to me, the study is very valid. It's a randomized trial, and I think it was done, at least presented, and also described in the JAMA paper as executed, I think, according to the standard of care and the standard practice in which you need to do a randomized controlled trial. It delivered a very precise effect estimate in a relevant patient population, thereby, to me, providing high quality evidence that FAST, as it was tested here, does not improve patient outcome. And that making decisions based on fast, on the other side, also doesn't hurt patients. It doesn't produce any harm. What now to do with that conclusion? And, well, if you're in doubt, you could start by reading the editorial that was published at the same time. And actually, I had to read it twice because initially I was not sure that the editorial was on the same paper that I was discussing. One of the lines at the end of the editorial says, the FAST trial should move hospitals firmly in the direction of implementing rapid AST. And that left me completely puzzled because this implementation will come with a price. Definitely. And there is no discernible benefit. So it will be very, very difficult to consider this to be a cost-effective approach. And I was walking out of that session and walking towards the exhibition of the conference, where I see the multi-billion industry telling us that everything we must do is create fast susceptibility testing and rapid diagnostics. I'm a senior now in this field, but this has been so for 30 years now. And this study, again, doesn't provide the evidence that we're really changing something for patients. And it could be, I think the study tells us, again, that gaining several hours in changing antibiotics towards what we consider optimal treatment does not yield the obvious gains for patients that we are expecting. That could mean that our empirical treatment is so good that changing that in a way that we think that makes it better doesn't make a difference. Or the other way around, that it's not really the antibiotic treatment that makes the patient outcome. That's more the underlying disease of the patient that determines whether the patient survives or not, how long that patient will stay in hospital. So I think this is a very, very important study that raises serious questions whether all our expensive approaches to fasten our practices related on susceptibility testing is really worthwhile. And I'm very anxious to hear what you think of this.
Well, for me, this is a really good example of why we need randomized controlled trials in general, because it seems obvious before doing the trial that if you have a technology that gives you the antimicrobial susceptibility results much faster and appropriate antibiotics faster, that would translate to benefit. And in fact, the old way of changing practice in infectious diseases, 10, 20 years ago, people would have just done studies based on, does it give you the results faster? Yes, therefore we should do it. But now the bar is higher, right? We're more and more experienced at doing trials. Now we want to see things actually change outcomes. And this hasn't changed outcomes, which was quite disappointing. But it's a good example for me of why we need randomized controlled trials.
Marc, I love that your interpretation is so subversive. I think
it's following the data. That's something else. That's something else than subversive.
You're completely
the editorial is subversive.
Yeah. I have to say, I fully agree with you. And I would maybe even question, you touched on this, Marc. Obviously, empiric therapy is going to be good, right? That's why we choose it. So maybe the outcomes, maybe the targets here are not the right ones, right? Maybe they don't have to ask that question, is this good for the individual patient? Maybe there could be such a thing as a randomized controlled trial that actually says, okay, is this good for the collective? Is this good in the aggregate? Because we do care about proper antibiotic use, right? I think they set themselves an impossibly high bar. How are you going to show that?
But the antibiotic resistance signal is interesting because the larger, let's say, the more extensive the resistance is, the larger the signal of an effect, which could lead to the interpretation. We just have to wait until there is more resistance, and then this will improve outcome. But we're not waiting. We're changing our empirical therapy for that. So if the resistance goes up, the empirical therapy will change as well. So either we wait until we have no more possible changes in empirical therapy, but then, of course, we probably also won't have much options to adjust. This trial will definitely not end the discussion and studies on how we diagnose and treat patients, but at least we have a new answer here. And with that, I would give the floor to Angela for the next trial.
Yes, I will be pivoting from diagnostics to treatment. I'm going to tell you about the CMI trial. And first off, I've got to thank Emily McDonald for the following notes on this trial. Full disclosure, she was meant to be co-hosting today, not me, but she ended up being on a flight right now somewhere over the Atlantic. So that's why I get to be here. She did not leave us hanging, though. You may recognize her in some of these notes in the description of the CMI trial. So the full title of this trial is Randomized Clinical Trial of Meropenem versus Cefmetozole for Definitive Treatment of Bloodstream Infections Due to ESBL Producing E. coli. First author on this trial is Kei Hayakawa, and the senior author is Yohei Doi. Another quick disclosure, this trial was presented only by poster, so our details are quite limited, and there, of course, was not this sort of presentation followed by discussion dialogue around its results. So here we go. The rationale for this trial, like the Peter Penn trial, whose interim results were presented this year at the trial run session and were also discussed in depth in our most recent episode, was to test a strategy to spare carbapenems, in this case for the treatment of ESBL, E. coli. And as we know, the Marino trial concluded that definitive treatment with piptazo, piperacillin tazobactam, was not non-inferior to meropenem for essentially ESBL bacteremia. The interim results of the Peter Penn trial do not have the same conclusion. Again, go back and listen to our most recent episode. But either way, we need alternate options to carbapenems. So enter the antibiotic cefmetozole, which has demonstrated in vitro activity against ESBL E. coli. And in observational studies, it has also shown outcomes similar to those with carbapenems. But of course, observational data are not enough. So I'm going to tell you a little bit about this antibiotic because possibly you may never have heard of it. It's only marketed in Japan, where it was developed, in China, South Korea, and Taiwan. It was actually approved in the US a long time ago, but it was withdrawn from that market in the 1990s, not for any safety signals, but simply for commercial reasons at that time. So cefmetozole is a semisynthetic, broad-spectrum cefamycin. It's often classified as a second-generation cephalosporin, and it's used parenterally only. It inhibits bacterial cell wall synthesis by targeting penicillin-binding proteins. In these 4 countries, it's used frequently for intra-abdominal infections, pelvic inflammatory disease, respiratory infections, and soft tissue infections. So you can guess that it has very broad-spectrum activity, gram-positives, gram-negatives, anaerobes, including Bacteroides fragilis. On the safety profile side, it can cause hypoprothrombemia, so it can prolong the prothrombin time and therewith predispose to bleeding. It's also reported to cause delirium, and finally, it can cause a disulfiram-like reaction, so that means no cocktails while you are having your ESBL bacteremia treated. Like other beta-lactams, there are dose adjustments to be done for reduced renal function. So, onto the trial now that you've had your cefmetozole primer. CEFMEC was a non-inferiority trial comparing cefmetozole to meropenem. This was an entirely investigator-initiated trial funded by Japan's Society for the Promotion of Science, which is essentially the equivalent of an NIH. So the study took place in 27 hospitals in Japan, and it was open label. Like most of these trials, patients were already on antibiotics at the time of enrollment, which could occur up to 72 hours after the index culture. Randomization was 1-to-1, and it was stratified by infection source. No further details available. Again, we're going off a poster. And by hospital site. Both drugs were prescribed as 1 gram IV every 8 hours, with each dose infused over, quote, 30 minutes or more. And the duration was determined by the prescriber, but was between 5 and 14 days. The pre-specified non-inferiority margin was 10% for a primary outcome of mortality, and a sample size of 196 was needed for 80% power. So in the end, 207 patients were randomized, but we don't actually know out of how many screened. That's not marked. There were different populations. There would be an ITT population, but we don't have those results. We are given the modified ITT1 population, which had 199 patients out of those 207 randomized. Among these 199, 102 were randomized to cefmetozole and 97 to meropenem. So these patients were older, median ages of 81 and 83 in the cefmetozole and mero group, respectively, and around 45% were male. About 60% of the infections were community acquired. A low percentage came from the ICU, around 10%, and the PIT score was low at 1. So many infections, just like in Peter Pan and Merino and Astarte, were urinary in origin. It's pretty classic for these gram-negative drugs. From the MITT1 population, 2 out of 102 patients died in the sifmedazole arm, and 5 of 97 patients died in the meropenem arm. So this translates to a risk difference of minus 3.2% for mortality, clearly making the 10% non-inferiority margin. There were several subgroup analyses, but the number of deaths were so few, it seems less relevant to get into them, as many of them even had 0 counts for the outcome. The investigators did look at retrospectively at a 5% non-inferiority margin, and the p-value for that was 0.005. On the safety side, there were very few serious adverse events, fewer than 10% per arm, and none of them were deemed due to the study drugs. So to conclude, this trial was quite small and had few events. Patients were older and comorbid, but certainly not critically ill. The investigators did successfully demonstrate non-inferiority of sifmedazole to meropenem with a 10% non-inferiority margin, and likely even with that 5% non-inferiority margin. So the trial has a lot of strengths. Obviously, it's beautifully randomized. It's nice, multi-center, 27 sites, and it uses a hard objective outcome of mortality. It, of course, has some weaknesses, the biggest one being its open label design, but I can tell you placebo-controlled trials are really hard to do on an investigator-initiated budget. And here, the investigators rightly chose essentially the most objective primary outcome possible. I would say right now, the other weaknesses are likely mostly related to reporting, which is really not the investigators' fault, as they were restricted to a poster. So I'm really looking forward to reading the full report. In the meantime, I would say it seems like we now have another study suggesting that definitive therapy for ESBL bacteremias may not always require a carbapenem, which is exciting for stewardship, of course. So while some of these alternative antibiotics that are being tested may not yet be available in all countries, results from randomized trials like these can be used to advocate to governing bodies for access, which is why we are giving this trial some airtime here. A little more access in other parts of the world would be good for everyone. Guys, what do you think?
I've got a comment that's not really about the trial, although I think it was a great trial, very interesting. I don't know if we're supposed to be saying this, a bit spicy. My children would say a bit of tea about this, but why was this given a poster? We had a major trial run session where we talked about ESBL gram-negative trials, both of which were either half finished or post hoc. This was finished and it showed something. So I kind of feel like apologizing to the investigators, but not that it was anything to do with me, but that this got given a poster. So now that I've got that off my chest, let's keep talking about the trial.
Oh my gosh. Okay, I say let's not cut this, and I will take the heat because the ESCMID Global Program Committee, which none of us are on, clearly doesn't agree with us. I do agree with you, Josh. My best guess, and again, I can't speak for them, I'm not on the program committee. My best guess is that they're just like, look, this is an antibiotic that's available in a very restricted part of the world. There has to be relevance, clinical relevance, and I would assume that that's where the thinking. What do you think?
But so is temacillin.
I was going to say, so I'd push back on that a lot because the antibiotic that was given the platform is only available in 2 countries or 3 countries currently, I think, which are very small.
You're talking about temacillin.
Yeah, it's only available, in Belgium, and no offense to Belgium, but I think Japan's bigger. So I think that it is a shame that this was a poster considering there was a session on ESBL bacteremia treatment. So kudos to the authors for this. And I think that would have loved to unpack this more if we knew more than just what's on the poster. So.
Ouch. Yeah. No, I mean, you're absolutely right. You're absolutely right. I don't know if the same subgroups judge all of this. I have no idea how the program committee is assessing these things. Tons of trials are coming in. I mean, ESCMID Global is spoilt for choice. As you were saying, Erin, they have so many trials to choose from. Little anecdote, by the way, our 2018 nitro versus phospho trial that had more than 500 participants over 3 countries, we were also given a poster initially. And we pushed back. We were like, this is an investigator-initiated, publicly funded trial that matters. And they allowed us to do an oral presentation. Moving on, Josh, you're up next.
Yeah, thanks. So the trial I'm going to present is called the COBRA trial. That was also presented in another late breaker session, which was focusing on surgical site infections. And quite a lot of people I've spoken to have identified this one as the trial that they think will change their practice. So this was looking at patients with acute cholangitis, and it was randomizing them to 1 day of antibiotics, that being the intervention, versus the current standard of between 4 and 7 days of antibiotics after drainage of their biliary tract in obstructed cholangitis. So there are guidelines that exist. For example, the International Tokyo guidelines, they recommend 4 to 7 days. I've checked the Australian guidelines are similar after a successful ERCP drainage of cholangitis. There was a randomized trial published in 2024 comparing 4 with 8 days, showing that 4 days was not inferior. And there's observational data suggesting that even shorter than 4 days is just as good. So that was why they did this trial. This was done in 31 sites across the Netherlands. It was an open label, government-funded, investigator-initiated trial. The patients had acute cholangitis. They had to have had ERCP-guided drainage of their biliary tract, which was generally obstructed by a stone, and they had to have had their fever resolved. They excluded patients with immunosuppression, neutropenia, Staph aureus bloodstream infection, Pseudomonas bloodstream infection, a liver abscess. And of note, they did not exclude people with bacteremia in general. So think about it, they're getting 1 day of antibiotics. Some of these patients had bacteremia, about 30% of them actually. They were randomized on 1-to-1 basis, and it was stratified on the causative organism, gram-positive, gram-negative. There was a blinded outcome assessment committee, and the primary endpoint was clinical cure, and that was defined as being symptom-free by day 14 and having no relapse or death by day 30. So it was a composite of those things. There were a number of other outcomes, including 90-day relapse and others. It was a non-inferiority trial with a margin set at 7.5% absolute risk difference. They based the sample size on a 90% expected cure rate in both groups, and that gave them a target sample size of 416. They end up having 410 patients randomized out of 745 screened, 205 in each group. The mean duration of antibiotics actually received was what they were randomized to get, so it was 1 day in the 1 day group and 5 days in the longer group. And the bottom line was that there was no difference between these two groups in the primary outcome. So it was 95.1% versus 93.7% in the short versus the long group. So that met the non-inferiority definition. The absolute risk difference was 1.5%, but it was approximately zero, basically. There were a number of subgroup analyses, and in these subgroups, the point estimates all favoured the shorter course, including those with gram-negative bacteremia. And in terms of the secondary outcomes, again, they all favoured the short course or were no different, and that included 90-day mortality. The only thing that was different was antibiotic-related adverse effects were more in the long group, so 16.6% had at least one antibiotic-related adverse effect in the long group versus 8.3% in the short group, and that was statistically significant on the Frequentis design that they used here. So for me, this was a really nicely done trial. It very clearly showed that 1 day of antibiotics was non-inferior to the current standard, and that it's kind of audacious to do that in patients with gram-negative bacteremia, but, it worked. To me, the key issue is around generalizability, because a lot of patients I see with acute cholangitis don't get an ERCP done. And if they do, they don't get it done quickly. So I didn't mention, but on average, the patients had their ERCP about 48 hours after admission to hospital. And often in my hospital system, they'll wait around for ages to get their ERCP or just not have one at all. And since I've seen this trial, I've been rounding on patients the last couple of weeks, and nearly none of the cholangitis patients I've seen would have been eligible. So the last 3 I've seen, for example, had underlying sclerosing cholangitis, and they just get kind of recurrent cholangitis without an obstruction. So I think if I do have the typical patient with a cholangitis and a stone that have an ERCP, it's unblocked and they're quickly getting better, I would stop their antibiotics just like we do for appendicitis as soon as their appendix is out. Another caveat is that this is not published yet, so we only know what was presented, but it has been submitted to a journal and the protocol has been published. So I look forward to seeing the full paper, but I think from what I've seen of this should change practice.
Yeah, I think this is incredible for two reasons. One, so I would say if patients are not bacteremic, as soon as their ERCP is performed, we stop antibiotics. So that tracks with our practice in that regard. We actually don't even give the day after. But if they're bacteremic, we give 7 days from source control. So this is, I think, immediately practice changing and wild to save patients up to 6 days. And I think, Angela, we've said this before on other podcasts. I think it goes to what we've been saying in that there's a lot of debate about oral step-down therapy for uncomplicated gram-negative bacteremia. Can you use a beta-lactam? Do you have to use a fluoroquinolone? And I actually think none of it matters. I actually think all of the oral step-down is a relevant exposure and that you really only need, I've been saying 3 days of therapy, like IV, 3 days of IV therapy for gram-negative bacteremia, but now maybe we only need 1 day from source control, which is insane, but also very cool. So kudos to the authors.
Yeah, I fully agree. I think this is exciting and we shouldn't extrapolate too much, but come on. I mean, this is hugely meaningful for abdominal infections where you have source control, where you know there are no remaining abscesses or, really this starts to change the discussion. This could be very paradigm changing.
And we know people from the Netherlands are awesome because we have two of the 5 of us. So kudos.
They're very bold. We love it.
They're very good at using not much antibiotics, people from the Netherlands. We need to learn from them.
Yeah.
I would make one minor note on the actual design trial, which, I think it's cool that they got away with this, but I wonder if reviewers will give them a hard time for choosing a non-inferiority margin of 7.5%. when that primary outcome includes mortality, right? And in the end, you have a very small event rate. With that non-inferiority margin of 7.5%, you're basically saying, if our event rate is 3%, we'll be fine with a mortality that ends up being double, right? It's always a game, right? You can't have a really narrow margin and actually afford the trial you want to do. But I wonder if they're going to have some trouble with that, honestly.
Yeah, I do agree with that, Angela. But on the other hand, it's pretty standard, even in mortality-based non-inferiority infection trials, to use 10% and even 20% some trials as non-inferiority margins. It's quite unusual to use what as clinicians, we think it should be, like 5% or less, because you end up getting a giant sample size then.
Nowadays, Josh, I think really even trialists don't honestly accept 20% margins. That's just
not 20,
just
but 10 is pretty the standard.
Still,
most commonly used.
Yeah, I see both sides. But anyway, we need to move on.
I'm going to hand over to Erin now, and she's going to tell us about the DOTS PK trial.
All right, DOTS PK, what a joy. I'm excited to present this trial. I do want to verbally say AbbVie, the company that owns dalbavancin, at least in the US market, I think globally as well, they have a database called the INFORM database. That's a gram-negative resistance surveillance database. And in 2025, I gave a talk at the Making a Difference in Infectious Diseases conference on the INFORM database, and AbbVie did pay my travel for that talk. So just to be upfront about my relationship with this company, I have an emotional relationship with dalbavancin and that I adore it. And so I will be frank in that this is something we've really shifted practice to in my institutions, because I think it's really wonderful for patients to not have to have a central line and get an antibiotic every single day. I think just bottom line up front, if you ask someone, hey, do you want this thing in your body and have to inject yourself all the time? Most of us would say no to that question. And so oral antibiotics, long-acting antibiotics, or things that help patients avoid that traditional standard care, I think is where we should be thinking in terms of the patient experience. And actually the SNAP trial team in New Zealand has like a really beautiful commentary on oral antibiotics in CID published last year by Walls et al. That has patient quotes about how they feel about taking oral antibiotics versus receiving IV antibiotics. If you guys haven't read that, it's a beautiful paper that really talks about the patient experience. So with no further ado, the DOTS pharmacokinetic data was presented by Tom Holland from Duke on behalf of the ARLG in the United States. The DOTS trial, for those who may not be familiar with it, was published in JAMA in September of 2025. This looked at dalbavancin versus standard care for complicated Staph aureus bacteremia. And they enrolled 200 patients in that trial, randomized 1-to-1, and they found a similar risk-benefit ratio with receiving dalbavancin or standard care. They said dalbavancin was non-inferior to standard IV therapy by clinical efficacy. I think it was 72 versus 73%. They did have, Josh, that 20% non-inferiority margin, though, and that was some very reasonable criticism of that trial, is that the non-inferiority margin was massive. You could like sail a ship through it. Probably too big. That is a significant clinical difference. But they had, similar efficacy in terms of percentages, similar safety. It notably was not their primary. They tried to power this for superiority, and it was not superior per a door analysis. So that's the background of the Dutch trial. But the cool thing is that they had a pre-planned secondary analysis of pharmacokinetics in patients that received dalbavancin. And their objectives were to characterize the pharmacokinetics of total and unbound dalbavancin in patients that had complicated Staph aureus bacteremia. And this is the first study of actually infected patients. We have lab data before this and model data, hollow fiber models. But as we always say, people are not hollow fiber infection models. So people matter. Your immune system matters. Your sickness impacts how you clear drug. So what did they do? They took plasma PK samples of total and unbound drug before the first dose, 10 minutes after the first dose, at 6, 12, and 24 hours, at day 8 before the second dose, and then day 22, 44, 70. They used a non-linear mixed effects model, and they applied that to simultaneously characterize total and unbound dalbavancin in terms of concentration time-dependent data. They wanted to identify patient factors associated with variability in dalbavancin exposures. Again, everyone's getting the same dose. Some patients got a little bit of a dose adjustment if they had renal dysfunction, but most people are getting 1,500 times 2. And they wanted to evaluate, here's the real important one, whether dalbavancin exposure was associated with clinical success. So I think this is awesome. We have animal and hollow fiber infection model data showing us that exposures out to 21 days after a dose of dalbavancin may be above a minimum inhibitory concentration of 8. And so that was reassuring, that lab data, because practically when you're trying to administer this to patients, not every patient can come back at exactly day 8 for an infusion appointment. And so those like operationally and, in my health system, those lab data gave us some relief that if a patient said, I can't come Monday, can I come Wednesday? We were like, yes, you're not going to die between Monday and Wednesday. You're going to have enough dalbavancin. There's some wiggle room in this like day 0, day 8 dosing regimen. But now we have people data, which is even better, right? And so again, before the Dutch trial, all the PK was based around this neutropenic murine thigh infection model, which was a 6-day model. And it really showed us that with these 2 doses, you should have therapeutic concentrations for 42 days of total and free drug. And that assumed a 93% protein binding. So what did they find in this PK analysis? So of the 100 patients in the trial that were randomized to dalbavancin, 97 of them had samples available, and then 93 patients were event-free through the first few weeks and evaluable at day 70. So this is the PK sampling from 93 patients. And they found that, one, we were wrong about the protein binding assumption. Dalbavancin is actually highly protein bound, greater than 99%. Over 90% of the samples had a fraction unbound of less than 1%. So this is significantly protein bound. Indeed, later unbound concentrations in the patients they sampled were undetectable. At day 42, 73% of the patients had a level below the limit of quantification for unbound concentration. And at day 70, 98% had a level below the limit of quantification for unbound concentrations. So super protein bound. Creatinine clearance had really no association with the fraction unbound, but albumin, as expected, had a modest inverse relationship with fraction unbound. So the lower your albumin, the more drug is free, the more drug is cleared, the more likely you are to potentially have underexposure, which is the story we see with cefazolin and ertapenem. This is increasingly becoming incredibly important in the pharmacotherapy space in assessing patients' albumin and then TDM accordingly. So here's the real important part to end with. The relationship between dalbavancin exposure and clinical success. Does it matter? I think this is the holy grail question for any antimicrobial. Is there a MIC exposure relationship? Is there exposure outcome relationship? And so among patients in the Dutch trial who experienced clinical success, dalbavancin concentrations were higher on average than patients who experienced failure in both their AUC exposure and their day 22 single point concentration. They saw a similar trend with unbound concentrations, and they found that efficacy increases as dalbavancin exposure increases when they divided patients into kind of these thirds. And so the highest level, like the highest chunk third, had the highest rates of efficacy, and then there was a middle group and then a lower group. And to take it one step further, they found that there was a cut point they could declare. So 32 micrograms per mil seemed to be this cutoff. They had 30 patients that had a drug concentration greater than 32 at day 22. Of those, 29 out of 30 experienced clinical success. Contrast to the 60 patients who had a drug concentration less than 32 at day 22. Of those, they had a 25% absolute lower success rate. Only 43 patients, or 68%, experienced clinical success. So what do we do with these data? I think, one, we know that dalbavancin is 99% protein bound, and albumin is going to matter when you're treating these patients. Higher exposures are equal to higher success, and they really didn't see any safety signals with the higher exposure, although the numbers are quite small when you're looking at 30 versus 60 patients. You're not going to get an AKI signal, but no notable glaring safety signal with a higher exposure. The parent DOTS trial, again, gave 2 doses because that was the data we had from in vitro models. And then there was also a small RCT in osteomyelitis that used 1,500 milligrams times 2 doses as kind of definitive therapy. So that's where the dosing regimen came from. But clinical failures in DOTS were relapses that occurred later. So the direct quote from the presenting author here was that it was biologically plausible that it is due to lower exposure at the end of therapy, these failures that they saw. So the real question is, we give 2 doses now, can we do better? And dalbavancin versus, standard Vanco or Dapto for 6 weeks was similar. Can we do better? Is there better than a 25% failure rate in Staph aureus bacteremia, which is kind of what we see consistently? So to me, honestly, these data were immediately practice changing in so much as I have 2 pharmacists that work for me that run a transitions of care program. And when patients are unhoused or have no insurance or significant barriers to receiving antibiotics outside of the hospital, we often are leveraging this agent to try to minimize the amount of appointments and resources they need. And we've just been doing 2 doses. And I literally texted my team from ESCMID Global and said, we need to start checking day 22 concentrations, which are fortunately available in the United States now. Those weren't available in the past couple of years. And so for countries that don't have this assay, I think you do need to pair this with TDM. Don't just blindly be given doses. We have a pretty clear cut point from these data. But I said we should start recommending day 22 concentrations. And then based on that concentration, potentially recommending more doses, patient-specific, infection-specific. So I think these are really cool data and interested in y'all's thoughts.
Yeah, I think it's super cool they measured the unbound because obviously we don't see that often. I'm just curious, what's the trend in the unbound concentrations? I haven't read the paper, but if it's linear between the unbound and bound, and if it's non-linear, then you would expect that the unbound is more critical to measure. In clinical practice, I don't see anyone measuring unbound. That's going to be really hard to do. But yeah, I would be curious to see if it's like a saturable situation or if it's a linear relationship between them.
I knew you'd be excited by this one, Erin. It's like a perfect one for PK people to nerd out about, right? But it's also cool that it's shown correlations between drug levels and clinical outcomes, which is really interesting. One of the questions this left me with, which I've brought up a few times over the last few years about dalbavancin, is what would they have found if they had used eretavancin in the trial rather than dalbavancin, which has a significantly longer half-life? I've got no conflict of interest here except emotional. I like eretavancin. That's what I use in our clinical practice here. We generally only give 1 dose rather than 2 doses, or possibly 3 doses now might be needed. So that's an unanswerable question because there's very few data, but it would be interesting to know.
It is a super good point because when we talk about the long-acting lipoglycopeptides, you have eretavancin on the table. Some people have the stewardship argument that eretavancin has activity against VRE and dalbavancin does not. So in a way, some of us save eretavancin for VRE, and it's like how we used to.
But do you actually use it for that?
Yeah, we will. So we have. You have less persistent VRE, but we've used eretavancin for particularly liver transplant patients, and once or twice we've done that Q48 hour for 3 doses lead in out of the MD Anderson. I think there's 2 case reports in total on this PK assessment. I don't know how accurate it is, but I think it just comes down to whatever drug you have on formulary. But it's kind of like the ceftobipril, ceftaroline argument. Now we have an RCT for one, no RCT for the other. Are they interchangeable? no. Like we know that drugs in the same class are not interchangeable, but we want to interchange them. You want to take whatever your formulary agent is, which honestly probably just comes down to whatever the best contracted price for your institution is, and then say like they're the same, but they're not the same drug. So it's important. We want ideally RCTs for all of the antibiotics. All right. Anne, you want to bring us home with the last trial of this first episode?
Yeah, I'm really excited to present this maribavir real-world data. So this was presented at the late-breaking research from the Lancet, New Interventions for More Established Pathogens, presented by Annalisa Pavlignati from Barcelona, Spain. So this trial was called Maribavir for Clinically Significant Cytomegalovirus Infection in Hematopoietic Cell Transplantation, a real-world retrospective international study of the Infectious Disease Working Party of EBMT. So maribavir was approved by EMA in Europe in November 2022 for treatment of RR or relapsed refractory CMV, infection or disease after solid organ or hematopoietic stem cell transplantation. It is shown limited toxicity, especially compared to the other drugs on the market, and approximately 10% resistance has been shown. So the study aimed to report clinical experience with maribavir in HCCT patients, and it's basically a retrospective registry study in children and adults who received maribavir post-stem cell transplantation after EMA approval. They included patients with a minimum of 12 weeks of follow-up and maribavir indication treatment of clinically significant CMV infection. And if used before authority approval, these were excluded. The primary endpoint was CMV resolution after 12 weeks after maribavir start. And secondary was the reason for discontinuation, overall survival, and no relapse mortality at 12 weeks since initiation. And response to therapy was basically defined as maribavir just discontinued with no need for additional anti-CMV treatment. And treatment failure was then defined as viremia progression, requiring therapy change, addition, viral rebound, etc. So they recruited 118 patients in 37 centers, but they do talk about courses. So from these patients, it was 126 courses, which majority were male and had acute leukemia. Only 9 of them were children, so mostly adults. Interestingly, 53% of patients received letermovir prophylaxis, which is kind of new practice in the last few years for this population. And the donor negative recipient positive CMV was 39% of the patients, so that would be our highest risk group. The donor positive recipient positive was 50% of patients. And the indication for maribavir was then either preemptive therapy for 19% of the population or second line preemptive therapy 48%. And reason to use maribavir was refractory of resistant CMV for 51%, 25% because of neutropenia, renal impairment for 12%, and other toxicity to previous antiviral agents was 10%. And as expected, the previous antiviral treatment was either valganciclovir for 40% or foscarnet 18%. Now we're going to look at some of the results. So either the response on preemptive therapy or during CMV disease. So response for the preemptive therapy courses in 109 courses was 81% was good response. So that was a very, very nice outcome. Failure was only 17%, 2% lost to follow-up, and basically no response failure was only 1%. But CMV disease during preemptive therapy was in 5%. So 5 patients, 5 courses. Response for CMV disease, that was in 17 courses. There was 70% response. 12% of these 17 courses had progression on maribavir, and 18% were lost to follow-up or not evaluable. Resistance was actually in the no response courses. The 20 courses was tested in only 6 courses, and from those only 2 had a resistance in UL97. The reason for continuation was then 76% was end of plan therapy. And overall survival at 12 weeks from maribavir start was 82% and no relapse mortality was 15%. So basically maribavir showed to be effective treatment option for clinically significant CMV in stem cell transplantation, high response rates with limited toxicity. So the authors and presenters suggested maribavir use beyond the EMA approved indications should be further explored. I really like to see this study because obviously it's real world experience with maribavir. We have used it very little actually. We always use it when our backs are against the wall. So when the patients are already really poorly and then we have lost those patients actually. So I've heard some practices that people start with foscarnet and then when the viral load is coming down, then they continue with maribavir. So that's all like anecdotal experience still. But I think this is a nice initial overview of maribavir use. It is retrospective and of course not a huge amount of patients, but I think it shows pretty promising results actually. I don't know if anyone else has some ideas on this.
And is this different to what was seen in the registrational trials of maribavir, the kind of response rates?
The registration trial was for refractory and resistant CMV as well. So it's the same indication. So they don't actually explore. They just kind of said, yeah, we should explore also maybe as a first-line treatment. I guess that's what they mean. I would be hesitant to do that actually, because I know ganciclovir is really effective.
Yeah, I mean, I have another emotional conflict of interest because I love CMV. It's my favorite.
Yeah, same.
It's my favorite infection. It's so great. It's so cool. The drugs are so neat. But the drugs are also terrible. And so I think maribavir coming to market was such a huge moment for patients. I think an oral effective therapy for a terrible infection was just so tremendous. So seeing it expand in patient populations and use is really exceptional. We save it too. I mean, maribavir has a very low barrier to resistance. You only need one genetic mutation to get resistance. And so it's not super great for patients with high viral loads and whatnot. So ganciclovir is still treatment of choice. So then if patients can't tolerate GAN or Valgan, we switch pretty readily. And then it's obviously become relatively first line for ganciclovir resistant CMV.
Yeah,
pretty
and actually
expensive.
it's expensive. Exactly. Yeah, it's expensive here as well.
You know what else is expensive though? Giving 7 electrolytes a day and having monitoring for IV foscarnet and ending up on ECMO and dialysis or whatever the heck we do to patients because we destroy their bodies by trying to give them foscarnet. So I think the cost effectiveness of maribavir is probably more than said itself.
Touché.
Yeah, the question is, should we start it earlier? Because now I feel like we give them like in the last resort. Here we see also very low resistance, so that's a good thing, I think. I also see that they have a good response, so I'm just curious, indeed, with high viral loads, what would be your outcome there? And also they actually had like kind of a low risk population you might debate. Like only 30% was the donor positive, recipient negative. So yeah.
Sounds like it's a perfect segue for a prospective randomized trial, right? I mean, we always have our little rule. Retrospective studies should not change practice, but they should be the sentinel, right? They should be a big glaring warning sign, in a good way warning, hopefully, saying, wait, we see something. We need to go further with our trial.
People are so hungry for this data. I saw like people just presenting case series with maribavir in the conference because there's just not enough real world data. So I'm really happy that this is out there now. We just don't have enough experience.
So in cell transplantation, it's flipped though. So your R positive is your high risk because the donor is the donation is the immune system. So while you can have CMV obviously in your entire body, but it's flipped. Right? Isn't that crazy? So it's flipped from solid organ, wherein recipient positivity is a higher risk for reactivation.
Oh well, then it's fine. Yeah, then they had a very good population, like half of them at least. Yeah.
The other interesting thing about cell transplantation is it's more often a reactivation than a primary infection, as opposed to a solid organ where that's different.
So all of them most of them are on PET, so preemptive therapy. That means that those viral loads are very low.
Yes.
Those viral loads are very, very low. They're like around 1,000, in the hundreds even.
Correct.
Sometimes.
Correct, yeah. And we don't use maribavir for high viral loads. But
no,
anyway,
exactly. So this whole population is going to, like largely, is not going to have a very high viral load, I expect.
Okay, so I think that brings us to the end of the first episode, right? These are the trials we were going to cover in this one. So I'll close off by saying thank you so much for the conversation. And thank you for listening to Communicable, the CMI Comms podcast. As mentioned, this was part 1 of a 2-part episode, and we'll be back with more trials soon. This episode was edited by Katie Hostetler-Oy, and the theme music was composed and conducted by Joseph McNade. Now, for the first time, this episode was not peer-reviewed. At our editors' meeting recently in Munich, after looking at all our past episodes and how they did, we discussed whether to continue with peer review or not. Given that there had been no findings of content errors or unstated conflicts of interest to date as a result of peer review, and also the worry we're creating unnecessary work for our peers, we decided to go forward without peer review. But if you feel strongly about this, please let us know, and you can write to Angela by email. This episode will be citable with a written summary referenced by a DOI in the next 8 weeks, and any published literature we've discussed today can be found in the show notes. You can subscribe to Communicable on Spotify, Apple, or wherever you get your podcasts, or you can find it on ESCMID's website for the CMI Comms Journal. Thanks for listening and for helping CMI Comms and ESCMID move the conversation in ID and clinical microbiology further along.
Ooh, great job. Oh my gosh. Guys, can everyone stay half an hour more? Because we think we can do this.
I was just going to say, can we sidebar for a minute about this? So I don't think we can get it done in 30 minutes, can we? There's
No, but we can do it in an hour. Yeah. Yeah. What do you guys
I don't know what.
I can.
Or we just schedule it. I'm good till 9:30. I do have a hard stop at 9:30 here, so I have 58 minutes. I think we can do it if we just don't have any extra commentary, but if you guys have to, I know it's late.
How about we skip the bit of all our intros and all that, because...
Yeah. Should we skip the... Oh, how sad. I love this question. The skydiving is a yes or no question.
Yeah,
that is a parachute question.
Okay. Yeah, let's yes or no. I'm just going to say welcome back to part 2. We're not going to do intros because you know us all. Yeah. And we're going to get right in. And then Angela, and then I might just ask the skydiving question and then literally just say yes or no. Don't comment on people.
Okay.
Don't like.
Yes.
Okay.
Also do the intros from the previous episode, right?
Okay.
Yeah.
Angela, you can also take the intros from the previous episode. You can cut them and put them here.
Yeah, exactly. Yeah.
Okay. All right. Let's rock and roll.